next speaker, Dr. Leandro Naves. Dr. Leandro Naves has led the development and commercialization of innovative genomic technologies while serving as president and CEO of Rapid Genomics, which was recently acquired by LGC Biosearch Technologies. Leandro has a broad experience applying genomics to plant and animal breeding, having assisted numerous breeding organizations in their adoption of high-throughput genotyping towards germplasm characterizations and genomic prediction. So today, Dr. Leandro will be sharing with us targeted high-throughput SNP and haplotype genotyping with the Flex-Seq XL technologies. I will then pass on to Dr. Leandro. The stage is yours.
Thank you, Joy. Appreciate it. Good morning, everyone. I hope most of you are doing well, having a good. Okay. So thank you, everyone, for watching this presentation. Today, I'll be talking about the method that we have developed and commercialized under the name of Flex-Seq XL. And this is a method that we use for high-throughput SNP and haplotype genotyping. It's in a nutshell, we've genotyped hundreds of different species and projects. So we have quite a bit of competence in the agrigenomics and industrial breeding applications. So when we're thinking about developing these products and these projects with you, we're very much focused. We have to be very accurate and reproducible. We, at the same time, try to be flexible because we understand that in a breeding program, you have different needs and different applications that you have to adjust. And obviously, speed is important. So we try to be fast in delivering solutions to you. So that's what I'm going to be talking about. We are located, our high throughput laboratory is located in Florida, in the United States. We receive samples from all over the world, and we're currently expanding our capacity to other sites. So if you watched the recent presentation from John, you might have noticed that we LGC, we have sites in Wisconsin, also in the United States, and in Berlin, in Germany. So we're expanding the capabilities to those two sites as well, so we can better serve the world footprint that we are covering here. The one thing that I want to mention is that you, as a customer of this project, you're going to have access to our customer portal and LIMS, so you can track in real time where your samples are in the process. So you can see them advancing through, you can see where your plates are. You can actually click and see the individual step of every single plate and where they are in the process. And you can always follow this on our Rapid Genomics application, the mobile app, that's available for both iOS and Android. So you can see in real time where your projects are and download some information associated with that. So that's our idea, to give transparency to the process and allow you to control this to serve your goals. I think John showed a slightly different version of the slide, but it really shows the breadth of expertise and solutions that we offer to you as customers. So we can start with a whole genome sequencing, where we can do, let's say, either low pass or high depth sequencing to generate millions of loci. And as Rapid Genomics, we also have this Capture-Seq method that allows us to target anywhere from 1,000 to 400,000 loci. And then we go next into the Flex-Seq XL, which is the topic of today's presentation, which we're focusing on this environment of up to 45,000 loci as mostly commercially available. And obviously, you have other solutions like being discussing today on the PCR side of the business. As you know well, on this higher density of genome characterization, you're going to be focusing more on pre-breeding applications. You're going to be focused on genomic germplasm characterizations, transgenic insert characterizations, and so on. And then as you advance your breeding population, and you're going to be focusing more on genomic prediction, parentage identification, and that's when you're going to be focusing more on the Flex-Seq XL. And I do want to mention that Flex-Seq XL is available as a service. We're also taking this as a kit to the market for some beta customers as well. And soon enough, it's going to be broadly available to all the community. But if you're interested, there's solutions for both service and kits. And the number of samples tends to increase as we reduce the density of the genotyping, as you know well, for your needs. So I think a little bit of the difference that you're going to see working with LGC, with biosearch technologies, is that we embrace the common challenges that we have in the plant and animal genomes. So naturally, you have repetitive DNA and large genomes. You also have very diverse and heterogeneous species in many cases. In a lot of situations as well, you have polyploids, mostly plants, and you have to deal with that high ploidy that you observe in certain species. And less common nowadays, but still present, you have a lot of incomplete or unavailable reference genomes. But if you really work through it, you're going to realize that we've worked hard to solve and embrace some of those solutions, to some of those challenges as we're creating solutions to those needs, right? So, for example, one of the solutions to your repetitive and very large genomes is that you can do targeted genotyping. So you can focus your genotyping on the regions that are important to you and how many regions are important to you. So that allows you to exclude all the complexity of the genome that you don't necessarily need to work with. And then to handle all the diversity that exists in your population, you need to have very effective probe design and chemistry that allows you to target these diverse germplasm without having constraints about having SNPs near your target site, for example. And similarly, if you want to attack the polyploidy, you also have to have chemistries that allow you to go after those different homologs that are present. And perhaps you can also take a haplotyping approach where you're using nearby SNPs to create localized haplotypes that allow you to infer dosage and understand better the germplasm. And we can have very flexible genotyping approaches and really work with you to develop genotyping, whether you're starting with a new species or a new program within your organization, or if you already have matured species that you've been working on. So we're really trying to be flexible in that sense. So I want to clarify very quickly here. I think a lot of you know, but it's important to distinguish the difference between SNP and haplotype genotyping. So when you're thinking about the SNPs, you're really talking about a single nucleotide polymorphism, right? So in the case of this, if we have this being the genome as a representation, we have a target region. You're going to have one region of that target that is polymorphic. And in this example, if you sequence 10x, you're going to see oftentimes the A allele and oftentimes the G allele. So that's a very simple concept. But what matters to us as breeders and population geneticists is that in this particular case, we can separate our populations in only 3 classes. So for example, whether you're working on plants or animals, if you're genotyping a single SNP, those samples can either be homozygous for one allele, homozygous for the other allele, or heterozygous. So you can only break your population in 3 groups. That's when you're looking at a single SNP, which is commonly done in some technologies. But because we're sequencing and we're actually identifying a broader region of the genome, we can actually identify nearby SNPs on that target. And then together, you can combine them into a haplotype, a localized microhaplotype for your population. And what that matters to you is that now when it comes time to separate, again, these populations with lots of samples, either animals or plants, depending on the diversity of the species, you can now separate them in more. And I'm just showing here an example of 4 alleles that you can pick up from this population. But oftentimes you can have 6, 8, 10 alleles in a population, depending on your diversity. So for example, you can have homozygous for the alleles on loci on locus 1. You can have homozygous for the T allele on locus 2, and so on and so forth, all the way to being homozygous for the other allele on locus 1 and 2. So that allows you to separate your population better, and that's thanks to the ability of sequencing a localized region. The topic of today's presentation is on the Flex-Seq itself, so I'm going to be focusing a little bit more on this technology and give you some examples of how we've been using this for different programs. So in a nutshell, for those of you who are more interested on the molecular biology concept of this method, if we have a target on the genome, and I'm showing here two separate targets, the left one and the right one. But again, this could be 1,000, this could be 5,000, this could be 20,000 targets on the genome. So we work with you to design probes that are going to hybridize on both sides of this targets. And we actually, different than many other technologies, we actually target both strands of the DNA. So we're going to be targeting the up strand and the lower strand of your DNA. So we're designing probes for both of them. We're hybridizing these probes to the genome, and we're complementing the region in between the probes so we can actually sequence them. So it's a very simple protocol where we then incorporate the indexes so we can identify those samples when we sequence them together in a lane. And then we provide you with data that allows you to differentiate every sample for those target regions. So that's in a nutshell the way Flex-Seq Excel methodology works. It's a very fast protocol, hence being very high throughput. And some of the advantages that we built this method, I already mentioned one, that is a very high throughput protocol. So we designed this from the ground up, thinking about doing very routine applications. It is sequencing based, so that again allows you to leverage more data than just a single nucleotide polymorphism. And it has very few enzymatic steps. For those of you who are interested in potentially adopting this and insourcing this as a kit, this can be done on one plate. You just keep adding reagents to that same plate to the DNA until you get to a sequenceable library. We also obviously mentioned that this is a targeted genotyping. So you're concentrating your resources where it matters. You're sequencing only what you want. And it's legacy data compatible. So if you already have a set of SNPs, either from your molecular sister selection program or from your GWAS or from other literature research that you've done, we can incorporate those regions into the panel for you. It's actually a very flexible genotyping technology. So that allows us to sequence diverse germplasms. It doesn't matter if the species has a lot of diversity. It doesn't matter if it's polyploid. So that really, it's a tunable approach that you can use for your needs. You can adjust your panels over time. So if in 6 months or a year, you found out new regions of the genome that are important to you, we can incorporate those regions to the assay. You don't need to wait for 1 year, 2 years, 3 years until you can incorporate those changes that you identify in your population. And obviously, it really adjusted. You can adjust that to your marker density. You're going to see some examples where you can go from 1,000 regions all the way maybe to 20, 30,000 regions of the genome. And lastly, this is a probe hybridization instead of a multiplex PCR approach. And that brings a lot of advantages to the data. So you tend to have more uniform data. This tolerates more diversity in the target region because, again, you're hybridizing for a longer period, and that allows you really to select the regions that you want. And you have a very specific and on-target data. And that's because you're doing one hybridization versus, let's say, 30 cycles of PCR, where every cycle you can introduce some errors to that process. So I'll show you some examples and some data quickly here, but happy to discuss more with you on a need basis. So I'm showing 3 specific panels to begin with. So we have a 1,000 marker panel, we have a 3,000 marker panel, and we have a 5,000 marker panel. And these are commercial projects that we've done. So these are real panels that we commercialize for certain customers. And for each one of those panels, I'm going to be showing the data for 3 samples, just so you can see the uniformity of the reaction. And then you can look at this plot. I have on the x-axis, I have the markers. So in this case, 3,000. In this case, nearly 5,000. In this case, nearly 1,000 targets. And on the y-axis, we have how much data was allocated towards each locus. So what you want to see is how homogeneous, how uniform, or how horizontal this data is on your panel. And you can see that for all these 3 panels, the data is quite horizontal. So it means that you're having a very even distribution of data throughout your targets. And that's what you want when you're genotyping a large population. You want to have that reproducibility of over and over genotyping the same targets with high confidence. And then I also want to show a few metrics that we usually run to compare projects. So the first one I already showed you is the uniformity. So you can see that for the most part, we're getting very high uniformity on these panels. And again, these are commercial, real examples that we gather from projects that we do. And the next one is the marker recovery. So if you target 1,000 regions, how many regions do you actually genotype? If you target 3,000, how many do you actually genotype, and so on? And you can see that for all these panels, we're above 98% of the targets that we're going after, we're actually genotyping. And then lastly, what's the percentage of the data that is allocated to the targets versus, spurious regions of the genome? And that's particularly important when you're adopting this as a kit, in-sourcing that technology in your lab. And you can see that we get 95 to 98% of the reads that we sequence being on target. So it's a very high quality data. And obviously, we also ask the question, what is the agreement of the data compared to other genotyping methods? And we have a very high agreement. So the data is uniform, and we have over 90% of the targets are agreeing between the genotyping with other methods, such as whole genome sequencing or microarray technologies. And we have a nearly identical sample-to-sample repeatability. So if you sample one sample and you sequence the same sample again, you're going to see 99% repeatability on the genotype calls. So it's very efficient on that. And lastly, on the genotyping side, I want to show you one example in maize. So in corn, we did genotype about 17,000 loci, but collectively, we're genotyping close to 30,000 regions of the DNA, or SNPs of the DNA. So that's what I was telling you about this idea of building haplotypes. And we selected this from industry-proven markers that we selected from different cultivars and landrace. And the marker this is available for you to access right now. So from you can select 17,000 loci, or you can select a subset of that. So if in your program you want, let's say, only 10,000 or 5,000, you can select from this set of markers that are available to you. They're very equally distributed throughout the genome. And as I mentioned before, the data is also very homogeneous and uniform. So you can see this being a very horizontal line on the graph. And as a last comment, we mentioned that we have this ability to pick up nearby SNPs and build a haplotype. I don't have a lot of time to expand on that, but I encourage you to think about that concept. So I'm going to put one slide here, which is a project that we've done in blueberries, in plants. And you can see that for this particular example, we built thousands of those, but I'm showing only one case, one locus, where we have two haplotypes very clearly distinguished. So you can see this, for those of you who are familiar with the IGV plots, you can see the top graph being one of the alleles that we sequenced, and the bottom plot being the alternative allele that we sequence for this particular species. So you can see there are even some indels on this particular allele. So that allows you to create dosage, create some multi-allelic markers, which becomes very powerful. So to finalize, that we offer very tailored genomic solutions for every stage and circumstance of your breeding program or research question. We're really very much focused on this applied, very high throughput industrial scale of genotyping. And then lastly, we really want to pair this with scientific expertise. So we want to help you focus on adding value to yourselves as partners. So with that, I'll take any questions if we have, or just thank you for watching the presentation.
All right, thanks very much, Leandro, for sharing on the Flex-Seq technology and the advantages of this chemistry to tackle the complex genomes in a more robust manner. So we do have questions here. So the first question is, can Flex-Seq handle high polyploidy samples such as wheat and strawberry?
Yes, we've done those species. For example, we also have projects with sugarcane, which is also a very, not only is a polyploid, but it's also a variable polyploid. So you can have some plants are octoploid, some plants are dodecaploid. So absolutely, and this idea of sequencing specific regions and having a haplotype really allows you to get some clean data for polyploids.
Thanks, Andrew. We have some more questions that pops up. I believe you mentioned about some of the panel that developed for one of the customers on the com. That do you offer any other standard marker panels? If so, which ones and how many markers and what are the markers based on?
Yeah, we have a range of species that we've done that we can provide you with a faster genotyping solution. So that's going to be, let's say, soy, cotton, pigs, cattle, maize, and many other crops and livestock. And the range of density varies quite a bit. So we tend to have panels that are up to 20,000 regions of the genome, ranging all the way from 1,000 or 500 loci. So you can really choose from this range of density.
Got you. Thank you. And the next question, do you have to avoid SNPs adjacent to the targets when designing probes?
No, we actually don't. We can embrace the diversity that your species has. So we can design targets even if there are SNPs around the target site, because that's one of the efficiency of the chemistry that we built, is that it tolerates a lot of diversity that's present on your samples.
Great, great. And the next one is asking, what is the maximum number of markers that can be determined using this Flex-Seq XL technology without modification to the standard protocol? And what is the highest number of markers that have been done?
Yeah, we've done up to about 45,000, 50,000 regions. That's like as high as customers approach this with. But obviously we can do more as well if needed. And so, but that's but off the shelf as we're currently doing, I would say up to 30,000 would be a safe 20 to 30,000 would be a safe range.
Great, thank you. And the last question I saw from here is, what is the time frame for a typical project that requires probe design? And so let's say from probe design all the way to results, usually how long does that take?
Yeah, usually it takes about 6 weeks, 6 to 8 weeks, depending on the complexity. But once that panel is developed, we can really genotype this on a fast pace and give you much faster turnaround times in the matter of a few weeks. And I do see one question about what bioinformatics tool we recommend for haplotype from SNPs. We can work with you we do have a tool to do that. There are a few other publicly available tools. Happy to take this offline if there's a need, but there are some options out there.
Great, great. Thanks, Leandro.